Next, we are going to hear from our lightning presenters. And these speakers were competitively chosen from the Research Day abstract submissions and will be receiving a monetary award. We're going to take questions after all 5 presentations are shared. I just want to thank our lightning presenters for recording their talks in advance. So we're going to be hearing brief research presentations from Hailey Feinzig, Saniya Khullar, Akshay Kohli, Lexi Nelson, and Dr. Adrian Johnson, who is a newly promoted assistant professor of medicine. So we'll hear from the presentations, and then we'll have an opportunity for questions from our lightning speakers afterwards.
Hi, my name is Hailey Feinzig, and today I will be presenting on my senior honors thesis project in the Saha Lab titled The Impact of APPC Terminal Editing on Endosomal Lysosomal Pathway Dysfunction in Down Syndrome Fibroblasts. The incidence of Alzheimer's disease AD in individuals with Down syndrome DS is significantly increased compared to the general population, and these individuals often experience a much earlier age of onset. This is largely due to a triplication of the APP gene, which codes for the amyloid precursor protein. Endosomal lysosomal pathway dysfunction, characterized by the enlargement of early endosomes, occurs early in the course of both AD and DS, and has been shown to contribute to the mechanisms of neurodegeneration. Endosomal enlargement is the earliest detectable manifestation of AD pathology and is present in DS-affected neurons by 28 weeks of gestation. You can see the presence of enlarged endosomes in the DS fibroblasts. In AD and DS, endosomal lysosomal deficits begin with an increase in endocytosis and deficits in endosomal maturation. This leads to lysosomal deacidification and reduced lysosomal proteolysis. Most lysosomal proteases have reduced activity in these more basic conditions, contributing to the wide array of endosomal lysosomal defects and subsequent neuropathological outcomes shown here. Beta CTFs, which are formed by the cleavage of APP by beta secretase, have been shown to play a crucial role in the development of enlarged early endosomes. APP beta CTFs are elevated in the brains of individuals with AD and DS, and overexpression of beta CTFs, but not alpha CTFs, leads to enlarged early endosomes in cultured human fibroblasts and PC12 cells, as shown here. Our lab recently collaborated on a study that utilized a CRISPR-Cas9-based strategy to manipulate the APP processing pathway. Editing the region of the APP gene coding for the extreme C terminus of the APP protein yields a truncated APP protein that is processed preferentially by alpha secretase, downregulating the production of A beta and beta CTFs and upregulating neuroprotective alpha CTF production. Using this genome editing strategy, we're looking at how editing the C terminus of APP impacts endosomal lysosomal pathway dysfunction in Down syndrome fibroblasts. Using EEA1 to stain for early endosomes, we will compare the size and abundance of enlarged endosomes in DS control and edited DS fibroblasts. Here you can see enlarged early endosomes in our DS fibroblasts that are not present in the control fibroblasts. We expect this editing of APP and DS fibroblasts will ameliorate this endosomal enlargement. Additionally, we are looking at the deacidification of lysosomes by measuring the activity of cathepsin B. Unlike most lysosomal proteases, cathepsin B shows an increased rate of proteolysis at higher pH. Cathepsin B functions as a beta secretase in the processing of APP and may contribute to increased production of A beta and beta CTFs. Additionally, inhibition of cathepsin B has been shown to improve memory and reduce A beta in an AD mouse model. We have found a significant difference in cathepsin B activity between DS and control fibroblasts, and we expect that APP editing in DS cells will result in a reduction of this activity. Finally, we plan to investigate the activity of other lysosomal proteases in DS and edited DS fibroblasts. Most lysosomal proteases have decreased activity at the higher pH that occurs in DS and AD associated lysosomal dysfunction, which leads to a buildup of waste within the cell. We expect that APP editing will restore the activity of these proteases near the level of activity observed in the control fibroblasts. And with that, I'd like to take a minute to thank those who have helped make this work possible. The Saha Lab, in particular, Dr. Saha and my graduate student mentor, Kirsten Gimsey, as well as our collaborator, Dr. Roy. Thank you all for listening.
Hi everyone, I am Saniya Khullar, a PhD candidate in biomedical data science. My presentation is on multi-omics gene regulatory networks linking risk SNPs and neuroimmunology to Alzheimer's phenotypes. Transcription factors, TFs, they bind to DNA sequence-specific TF binding sites near the enhancer or promoter of the target gene to help regulate the expression of that gene. This TF has a motif TACG, that binds to this TF binding site and helps regulate the expression of this target gene. Individuals who have the single nucleotide polymorphism, this SNP, this mutation in one DNA base, having an A instead of a G here, this TF cannot bind to this binding site and this target gene may be dysregulated. Our understanding of the cellular and molecular mechanisms from disease risk variants to Alzheimer's disease phenotypes is still limited. Most SNPs are on non-coding regions, and it is difficult to link them to genes and then to functions. In response, we built a computational pipeline, AD-SNP-Pheno, to help uncover gene regulatory networks from AD risk SNPs to genes and phenotypes. AD-SNP-Pheno inputs multi-omics data. In the end, we output the regulatory network linking SNPs, transcription factors, regulatory elements like TF binding sites to target genes for AD biological pathways and phenotypes. We focus on these three AD-related brain regions, the hippocampus CA1, lateral temporal lobe, and dorsolateral prefrontal cortex, DLPFC. So these are some of the metrics that we found for our SNP to TF to target gene regulatory networks. 268 AD SNPs play a role here, and there are 50 common target genes that are dysregulated across these three brain regions. In the lateral temporal lobe, we identified 194 target genes with disrupted regulation due to AD risk SNPs. So for simplicity, to avoid hairballs, I selected these 15 target genes for this visualization here on the right. So green are SNPs, yellow are transcription factors, and red are these target genes. And you can see how these networks are connected, as well as some differences in how SNPs can have effects that are very complicated. KCNN4 is a target gene in this network. So I'm going to zoom in on this gene next and explain how to interpret this very key story that we uncovered from our networks, which I think is an important example of what we are trying to achieve here. So this network here shows disrupted regulation of KCNN4, a potassium channel gene, which is overexpressed in AD brains and associated with neuroinflammation. So KCNN4 belongs to a Braak progression gene module in the hippocampus and to an AD lateral temporal lobe that has a lot of biological enrichments. So SNPs break TF binding sites that disrupt FOXC2 and PAO2F2 regulation of KCNN4 in the hippocampus and E2F7 regulation of KCNN4 in the lateral temporal lobe. So these non-coding mutations impact KCNN4 regulation, which may help researchers better understand why KCNN4 is a key drug target for AD, even though no disease-related mutations have been found in the gene itself. We wanted to prioritize some of the extensive SNP to TF to TG relationships we uncovered, and thus we focus on flagging potential biomarkers for AD neuroinflammation. That may begin decades before AD clinical onset, so we use COVID-19 as a proxy for immune dysregulation and found shared pathways involved in both COVID and AD, such as the NF-κB pathway. A key AD hypothesis believes that AD is primarily caused by impaired NF-κB pathway signaling, where NF-κB TFs are activated, resulting in neuroinflammation, long-term potentiation, a decrease, and neuronal death. And COVID-19 NF-κB TFs activate cytokines, contributing to the exaggerated inflammatory responses seen in severe COVID. Looking at AD and COVID PEG pathways, we found that there are 22 shared genes between them. So then we looked at our original TF to target gene regulatory networks, whether or not they are disrupted by SNPs. And we looked at TFs that regulate these 22 shared genes, as well as potential target genes for these. And these are the total number of genes that we got across the 3 brain regions, and this is overall combined for the 3 brain regions. We also gathered 18 benchmark COVID-19 genes from 4 published studies. So we had these 5 gene lists, 4 lists for our models, and the last is the benchmark. So we use machine learning techniques to select the top 16 genes from each of the 5 COVID-19 gene lists to classify COVID severity in COVID patients using blood gene expression data for COVID positive patients. All 4 of our AD-COVID GRN-based models outperform the benchmark by having a higher area under the curve and accuracy and higher net benefit values based on decision curve analysis for every single probability threshold. I am happy to say we found 51 potential AD neuroinflammatory risk genes from our 4 AD-COVID GRN-based models, which may be helpful biomarkers for earlier detection of AD. Follow-up tests are needed, and we mapped our predictive genes back to our original gene regulatory networks to see how they are connected and flag SNPs associated with those core genes. So on the right is a network that shows how genes in the final machine learning model for the DLPFC interact with each other and with some of the shared AD-COVID pathway genes. A huge thank you for the support from the NIH, NIA, CIBM, Professor Daifeng Wang's lab, my committee, UW-Madison faculty and staff, my family. I also have an educational YouTube channel. Please let me know all questions. Thank you so much.
Hi, my name is Akshay Kohli, and I'm a graduate student in the lab of Dr. Barbara Bendlin. Today, I'll be sharing an analysis assessing the relationships between cortical myelin and cognitive tests from participants enrolled in the Alzheimer's Disease Connectome project. For some background, our interest in cortical myelin stems from some post-mortem observations made by Heiko and Eva Brock in the 1990s, where they made the link that development and maturation of cortical myelin occurs in the inverse pattern of Alzheimer's disease-related neurodegeneration. Regions such as the temporal and frontal cortices tend to myelinate later in life and are typically the regions earliest affected in AD. Within our own group, we've explored the role of intracortical myelin in aging in AD using MPN-RAGE-derived R1 mapping. MPN-RAGE is a powerful T1-weighted MRI sequence developed by Dr. Stephen Keksometti at the Weissman Center, which allows for the precise estimation of R1, a metric highly sensitive to brain myelin. We found that older individuals exhibited higher R1 or greater myelin content throughout the brain, and these results were strongest in the temporal and frontal cortices. We then investigated whether levels of cortical myelin varied in individuals with MCI and dementia. Here we have average surface maps representing the distribution of R1 in the cortex for our unimpaired group, as well as our MCI and dementia groups. Even just visually, you can see higher R1 in the MCI and dementia groups compared to the unimpaired group, and specifically within the temporal lobe. Given the strong postmortem observations made by Brock and Brock, as well as work from our own lab, we wanted to dive a bit deeper into understanding the role of cortical myelin in cognition. We asked the question, how are levels of cortical myelin associated with performance on tests of executive function, working memory, and processing speed? We hypothesized, based on our prior findings, that lower cortical myelin, as indexed by R1 mapping, would be associated with better performance on cognitive tests. In order to test this, we used data collected from 143 participants enrolled in the Alzheimer's Disease Connectome project. ADCP was a multi-site study with visits conducted at UW-Madison and the Medical College of Wisconsin. Participants underwent a variety of imaging, cognitive, and laboratory testing as a part of ADCP. However, for this analysis, we included participants that underwent the MPN-RAGE MRI scan, as well as completed the NIH Toolbox Cognitive Battery. We used the dimensional change card sort test as our measure of executive function, the list sorting test as our measure of working memory, and the pattern comparison test as our measure of processing speed. For our image processing, we input the T1-weighted MPN-RAGE image through FreeSurfer to estimate the peel surface and gray matter-white matter boundaries. We sampled R1 at the midpoint between those surfaces and averaged R1 in composite regions of interest created from the Deskin-Killeney Atlas. These regions included the anterior cingulate, frontal, parietal, posterior cingulate, and temporal ROIs. We used multiple regression for this analysis with 15 total models estimated. We included age, sex, and education as covariates, and we were interested in the main effect of regional R1 on cognitive test performance using the uncorrected standard score for each test. Within the executive function domain, we found that lower posterior cingulate and temporal R1 were associated with better performance on the dimensional change card sort test. With regards to working memory, again, we found a negative association between temporal R1 and performance on the list sorting test. And then finally, there were no significant associations between regional cortical R1 and processing speed. So our hypothesis was partially supported in that we found lower R1 was associated with better executive function and working memory, though there was no detectable relationship with processing speed. So we're consistently seeing that less cortical myelin is associated with better cognition. Why are we seeing this? In turning to the literature, a couple studies may help us understand what is occurring in aging and neurodegeneration. A study of cortical myelin in a multiple sclerosis mouse model found that when myelin was damaged in the cortex, they found that not only did oligodendrocytes act to repair the demyelinated axons, those oligos actually myelinated previously unmyelinated axons, indicating that during the disease process, the composition and organization of cortical myelin may change, potentially leading to altered cognitive ability. Another study focused on Alzheimer's disease in humans reported that when they stained myelin in the cortex, as can be seen in this figure, the control subject had clean raster-like traversal of axons throughout the layers of the cortex. In comparison, the late onset AD and especially the early onset AD participant had increased disorganization of myelin throughout the cortex. It's possible we may be detecting this increased disorganization of myelin using our methods. Thank you to all the co-authors for their help with this work, and especially Dr. Bendlin for supporting me throughout grad school. Thank you to my friends and family, the funding sources, and most importantly, all the research participants and their families who dedicate their time for this study. Thank you for listening.
Hi, I'm Lexi Nelson, a senior here at UW-Madison. I'm majoring in neurobiology and psychology, and I'm an undergraduate researcher in the Okonkwo Lab. Today, I'm here to talk to you about the association of vascular endothelial growth factor and vascular resistance in individuals at risk for Alzheimer's disease. So understanding vascular dysfunction and the role of cerebrovascular health in preclinical AD may be essential for developing interventions to mitigate its advancement. That being said, pulsatility index is a measure of vascular compliance that reflects arterial stiffening, and this stiffening has actually been postulated to reduce perivascular drainage, leading to amyloid beta accumulation, which, as we know, is a hallmark of AD pathogenesis. So a paper published by Dr. Rivera actually highlighted that individuals with Alzheimer's disease actually had higher pulsatility index when compared to controls, as pictured to the left here. And on the other hand, vascular endothelial growth factor is crucial for promoting angiogenesis. And some studies have established a relationship between VEGF and risk for AD, but there's conflicting evidence in the literature here. So although we have significant associations established for both of these factors, the relationship between them still remains unclear. So the primary goal of this project was to examine the association between plasma VEGF levels and cerebral vessel pulsatility index. So we procured 148 cognitively unimpaired individuals from the WADRC and RAP studies. Plasma VEGF levels were collected via blood draw, and 4D flow brain MRI was used to determine pulsatility index in the basilar artery, the middle cerebral arteries, the inferior and superior portions of the internal carotid arteries, the posterior cerebral arteries, the straight sinus, superior sagittal sinus, and lastly, the transverse sinus. We then ran multiple linear regressions and controlled for ApoE status, sex, age at VEGF sample collection, and then the difference in time between that VEGF collection and MRI acquisition. So our sample was 64% ApoE positive, which is roughly 3 times more than the general population, which gives evidence that our sample is enriched for AD risk. And 63% of that same sample was female. The mean age was 64.6 at MRI and 63.3 at the time of blood draw, showing that there wasn't a huge difference in the time of acquisition of those 2 samples. And then in the table to the right here, you can see that we found significant positive associations in the ICA, the BA, the PCA, and the TS. We also found a trend In the superior sagittal sinus, that was very close to significance. We also found no significant associations in the PICA, MCA, or SS. It's also important to note here that these data have not undergone any outlier sensitivity analysis. So this project reaffirms the importance of establishing and analyzing early vascular alterations, such as VEGF and PI, before potential cognitive impairment. These VEGF levels could potentially be used in risk calculations for AD, and in future studies, it would definitely be beneficial to investigate whether the associations found here are confined to a subset of the population. And sensitivity analyses to determine true outliers would also be very beneficial, along with increasing the sample size to better clarify our associations. But overall, we found in a late middle-age cohort enriched for AD risk, there is a significant and positive association between plasma VEGF levels and cerebral vessel pulsatility index. Thank you all so much, and a huge special thank you to all my co-authors and contributors. I could not have done this project without you, and I am so grateful for all of you. Thank you.
Hello, my name is Adrian Johnson. I am an assistant professor at the University of Wisconsin School of Medicine Public Health, Department of Medicine, and the Center for Tobacco Research and Intervention. As the title of my talk alludes to, I would like to shift our focus to a more broad definition of dementia research within the lens of public health, focusing on ways that we can prevent dementia in an aging population. An estimated 6.2 million people suffer from dementia in the US, resulting in an annual cost of over $350 billion. And that incidence of dementia is expected to triple by year 2050. Due to the rapid growth, overwhelming impact, and refractoriness of this disease to current treatments, greater focus is needed on targeting modifiable risk factors to prevent and slow the development of dementia. Targeting these risk factors could prevent approximately 40% of dementia cases. One such risk factor is cigarette smoking. Now, I'd like to acknowledge that there are controversies about the pathway through which cigarette smoking impacts dementia, whether it is indirectly through the 5 known risk factors for dementia or directly. It is inarguable that smoking cessation is an actionable and existing step through which we can effectively reduce the risk of dementia. Importantly, smoking is particularly problematic among older adults and is considered a late life risk factor. Currently, there are 18 million adults ages 45 and older who smoke. Unfortunately, older adult smokers are half as likely to try to quit smoking compared to younger adult smokers. And given the predicted doubling of older adults in the next 25 years, that 18 million, unless they quit, will soon turn into 36 million smokers. And given that older adults are disproportionately burdened by the negative health effects of smoking, this means more than double of all the health effects, medical costs, and deaths. Now more than ever, it's imperative that we help these smokers quit. Thankfully, when older adults do quit and use evidence-based treatment, they are more successful at staying quit. The problem is not, therefore, treatment efficacy, but a failure to motivate cessation attempts and the use of evidence-based smoking treatment. Therefore, the goal of this study is to develop a motivational message for older adult smokers to encourage both a cessation or quit attempt and use of evidence-based treatment during these attempts. For this study, we recruited 24 current cigarette smokers between the ages of 50 and 75 years old. They needed to have no diagnosis of dementia or mild cognitive impairment, and they were primarily cisgender females as well as Caucasian, and the smokers reported on average 3/4 of a pack per day. This was conducted via telephone with a semi-structured qualitative interview as well as a survey. And in terms of the analytic process, we used a directed content analysis of the transcriptions using a rapid analytic technique. The most commonly reported health-related concern was dementia, as well as cognitive loss and loss of functioning. And this was followed by some of the known other health effects of smoking. Most participants believed that they were at risk for their feared health consequence and that quitting would help. And although participants had seen a previous advertisement for smoking, the majority didn't feel motivated to quit by these existing advertisements. So when we gave them information about dementia, half of participants were unaware of any link between smoking and dementia. And of the half who said, yeah, we know something, really only a quarter of all participants remembered specifics about this link. The vast majority did find this message about smoking raising a dementia risk and quitting as a means to reduce this risk as motivating for cessation. And over half of participants stated this type of message would be more motivating than previous cessation advertisements. There were varied preferences for the exact content of the messages, but two broad categories arose: a hope, or what do I get from quitting message, as well as a fear, or what will I get if I keep smoking message. In terms of treatments or evidence-based treatments for smoking cessation, most participants were aware of smoking cessation pharmacotherapies from previous use, but their negative side effects, cessation failure, and costs were barriers to future use. However, the majority said they would be willing to use some cessation pharmacotherapy. And many had not considered cessation counseling and were unaware of how to access it, but still half were willing to use this treatment form. In conclusion, a hopeful as well as a fear-based message around smoking and dementia could motivate cessation for these older smokers. And although dementia is an important health consequence of smoking, older smokers are unaware of this modifiable risk factor. I'd like to thank you so much for your time and to share that I'm completing a culturally specific message funded by the ADRC Developmental Project, which we just wrapped up collecting data for older adult African American smokers. Thank you.
I would like to invite our audience, if you have any questions for any of our presenters, please make your way to the microphone and ask your questions. You also may ask questions of each other if you have any questions.
Hi, great job, all of you. My question was for Akshay. So you showed changes in R1 across the cortex associated with myelin. I was wondering if you showed a bunch of different areas, frontal cortex, temporal, et cetera. I was wondering if any areas stood out in relation to both changes in R1 and cognition.
That's a good question. That's a great question. So primarily over the different analyses that I've done for my dissertation project, the temporal and frontal cortices show the most change and most associations with age, cognition, and stuff that I didn't show with amyloid pathology. Another area is the inferior parietal lobule, which has been shown in prior studies of cortical myelin as well. I'm not too sure as to why that area is popping out, but that's definitely something to look forward to. But yeah.
Did you look at any subcortical regions?
Not in this analysis, but I have in the past. And with that as well, the entorhinal and perihippocampal white matter tend to be the most affected by aging and Alzheimer's pathology. Yeah.
Cool, thank you.
Hi, so I have a question about the last talk. So smoking is linked to dementia. Is there any evidence that like vaping is also linked to dementia? Because is it a nicotinic thing, or does it have to do with all the other junk that's in like a cigarette? Because obviously if smoking cessation is important, it's probably also important to prevent young people from vaping as well. So I'm just curious.
That's a great question. So there's not any research on vaping and dementia. There's very limited research, but I shouldn't say. There's older, and I'm trying to revive the area of smoking and dementia in terms of cigarette smoking. You are right that cigarettes have over 7,000 chemicals in them. Nicotine is the main addictive ingredient, and nicotine alone is actually a stimulant. So there is some work as transdermal nicotine patches and other ways in order to improve cognition. You've hit on a very hot topic area for the tobacco world, the nicotine world, in terms of, is it beneficial to prevent young people, not even adults, to start vaping? The answer is yes. There's also the other side in terms of harm reduction of older adults who are finding it hard to quit smoking. We do know that there are less chemicals in e-cigarettes, although the evolution of e-cigarettes is extremely rapid. It's a multi-billion dollar industry. So there's a camp that's very focused on using e-cigarettes to help smoking cessation. So your point of, is it important to prevent young people from using it? Yes. Will dementia prevention be the reason for them? I doubt it. So that's something to keep in mind too. Thanks.
Thank you.
I also have a question for you. Sorry, I know you just walked back. So for the older individuals who do quit, do you know if one of their reasons for quitting is because they're aware of the connection between smoking and dementia?
I would imagine no, given our findings where there was such a limited awareness in a currently smoking population of dementia. I would imagine that's not the case. Unfortunately, in this country in particular, that information hasn't been spread. In the UK, it has been used as a smoking cessation campaign, and particularly for the older adult population, we're thinking it could be a way in. I do want to highlight that for different cultures, particularly African Americans who we've started to work with the notion that you may be smoking for different reasons, and different reasons may motivate you to stop smoking. It seems very preliminary analyses. It seems that dementia is no more important than any other health consequence of smoking in that population. So I think that it's important to keep in mind who you're talking to, that everybody's different.
Thank you.
Yeah.
Hi, great set of talks. I also have a question for Dr. Johnson, and actually fairly similar to what Kelly was asking, but could you speak a little bit about the evidence-based clinical therapy techniques and why it is that maybe younger adults have a harder time adhering to that? Does that relate at all to the technique, or is that more?
So there's pharmacotherapy, which are the 7 FDA-approved medications, which is nicotine replacement therapy and 2 non-nicotine pills. And then there's smoking cessation counseling. And in terms of the primary orientations, I'm going to go into a bit of a detail rabbit hole.
That's what I'm looking for.
Cognitive behavioral therapy, and acceptance and commitment therapy are the most evidence-based. It's less so, as far as we know, an issue with adherence to the treatment, meaning going to the sessions, doing the homework, because CBT and ACT are very homework-based. It's more so that they have less success in doing it. So it could be an adherence piece, and that's very possible, and maybe an area for some mediation analyses. But I don't think that we should say that it's because they're not doing it. We just know that when older adults do it and make the decision to do it, they're more successful at quitting and staying quit. So this is even after they stopped the counseling.
Very interesting. Thank you.
Yes.
Sorry, I'm going to be annoying and ask another question. This is for Akshay. So I'm really interested in the new methods that you guys are using for looking at white matter and Alzheimer's. So you said more myelin leads to worse cognition. And so before I ask my sort of like out there question, you were looking only in the white matter, right? It wasn't any gray matter.
So this was specifically in the cortex. And if you look in white matter, it's the opposite relationship.
Okay, great. So it seems like it's aberrant myelination within the gray matter, and then in the white matter, you're kind of like flipping them, is essentially what you're saying?
Yeah.
So do you think it's, I'm going to ask you to speculate.
Sure.
Because, there's evidence that interneurons are very thinly myelinated, whereas obviously those larger projection neurons have really thick myelination. And, I don't know what the resolution is like on these MRI techniques because I've never done them. Do you think it's possible that these inhibitory neurons have such thin myelination that you're not detecting it initially, but then over time in these cases where the myelin flips, it's that the insulation or the myelin sheath is getting thicker around these interneurons, and so it's causing maybe some sort of weird overinhibition or something like that?
That is spot on in terms of how I'm thinking about it. I think there's another aspect to think about, which is cortical thinning that's occurring. So the sequence that we're using is 1 millimeter in resolution. And so in certain regions of the brain, in the cortex, you have like 3 to 4 millimeters in thickness, but areas like the motor and visual cortices are 1 millimeter on occasion. So we might be picking up just some compaction of the tissue as well, which is we're taking sort of an averaged signal of R1 in a given voxel, which is if it's 1 millimeter, you're just capturing whatever's going on in that section. So it could be a mix of that, but I do think that your kind of speculation on that is how I'm thinking about it as well. There are studies that show that a majority of the myelin in the cortex is encapsulating those inhibitory neurons. And so that might be what we're seeing.
So as a follow-up, do you guys have any plans to do like postmortem staining on any of this to look at, what the myelin looks like?
Akshay would like to graduate, so he will not be taking on that project, but we will have an opening in the lab. So we actually are at time. Thank you so much for those great questions. And if you have questions for our other speakers, I encourage you to find them after this session. I'd like to ask Dr. Astana to come now to the podium, and we will be presenting our lightning presenters with their certificates and awards. So when I say your name, you can come forward and retrieve your certificate. So our first awardee is Hailey Feinzig. Saniya Khullar, please. Akshay Kohli. Lexi Nelson. And Dr. Johnson. And we'll now take a picture together, and then we'll move on